So, welcome everybody. I'm David Wallace. I'm the director of the Newton Institute, and I'm really pleased to see such a great turnout for this workshop. It's a good chance for me, actually. I mean, this is the final week of a program which has been going on now 4 or 5 months. It's a great chance for me, actually, to give thanks to the organisers, to Mike and to Vincent, to Daniel, who isn't here, who's done a great job in organising what for me has been a really interesting program. I mean, my background's in theoretical physics and so forth, so I've learned a hell of a lot at this program, although not as much perhaps as I should have. But anyway, thank you all very much, and thanks as well to Katarina, who's been helping to organise this particular workshop. I can see from the program here you've got a fantastic week ahead of you, so I think I should just step down and let you get on with it. Thank you all again very much for coming, and I hope that you have a really good time with it in Cambridge. Thank you.
Just on behalf of the organisers, I'd like to welcome you here. It's, I guess, the end of a 4-month program for some of us, and it's been really interesting time. The program, there's a lot of talks, and we've scheduled some things called discussion, but what we intend those really to be is not formal discussions, but just an opportunity to chat over a coffee and catch up with old buddies and so on. We thought it'd be nice to have a photo, a conference photo, and we'll do that before lunch. We'll either do it today or tomorrow. We'll just see that most of the people, if most of the people are here by lunchtime, we'll do it today, otherwise we'll do it tomorrow. There's been 1 or 2 people who've been delayed because of these snowstorms across the Atlantic, and so we're going to have a slightly longer than usual lunchtime tomorrow. I hope you don't object to that. I think that's all really. If you have any questions, please ask either email Vince, myself, or Daniel, or Kathy, or the very helpful Newton Institute staff such as Tracy. Okay, well, I'd like to now bring on Vince, who's going to chair the morning session.
Thanks very much, Mike. So, first speaker from the conference will be Tandy Warnow. She's going to tell us about the Tree of Life, some algorithmic and software challenges. Thanks very much, Tandy.
Put this on.
Ah, yes, that's right. Maybe I should do that again.
Thanks, Tandy.
Thank you. Okay, it's my pleasure to be here. Is this on? I hope so. I gave a somewhat similar talk the first week of the session, that is to say, back in September, and so this is a little bit different, but a little bit overlap for those of you who are here. The first time you will find some part of this repetitive. My apologies. So, first thing is, how did life evolve on Earth? That's the basic scientific question we're trying to answer. There are lots of reasons to try to do this, including just fundamentally, it's an interesting question, how did life evolve on Earth, and what is the order of speciation events? But it has applications in biology in almost every possible way, including the design of drugs, understanding the origins and migrations of humans, various applications. Anytime you do protein structure prediction, protein function prediction, multiple alignment, you're going to have a phylogeny in mind. So having a phylogeny, having an evolutionary perspective, informs everything in biology. The Cypress project is a very large group of people, of about 40 or 50 people at this point, from something like 18 institutions in the US, plus some foreign collaborators, working on various aspects of the computational issues involved with estimating large phylogenies, including fundamental mathematical issues, fundamental statistical issues, databases, algorithm development, software library development, museum exhibits, etc. Just a lot of things going on there. A big part of it that's been happening is the algorithms component in the fundamental mathematics, which includes many different topics, starting with coming up with better heuristics for solving the major and hard optimization problems, but also including things like, given a large collection of trees, how do you store that set efficiently so that you can query it? How do you detect and represent horizontal gene transfers? How do you deal with phylogenies when what you're working with are whole genomes and you're looking at how genes are being reorganized within the genomes through rearrangement events? All sorts of different questions have been addressed, which is not to say we've answered them, but we have started to make some progress on them. The Cypress software distribution, which is open source and downloadable, also has a portal version. So if you have a large data set and you'd like to analyze it, you can actually upload it to the Cypress portal and get a very fast maximum likelihood analysis or a fast maximum parsimony analysis. We're also going to be adding POI, which does simultaneous estimation of trees and alignments, and we'll be adding Mr. Bayes as well. And we'll be adding other things as time goes on. So that is a big component of the work, is enabling analyses by systematists of their large data sets. Now, why do we keep talking about large data sets? Well, to start with, there are many interesting large data sets, including the tree of life. But there are data sets that other people want to analyze that are not on the scale of the tree of life, they're still large. So large could be, a few thousand, doesn't have to be a few million. Large could even be just, 100 sequences if you're trying to do something particularly challenging. So there are many data sets where the data set size makes it difficult to analyze. And those are the ones that we are trying to look at. The question is, why do you have to look at a large data set? Why not just look at all the 4 taxon subsets? Part of the answer for that is that taxon sampling, dense taxon sampling, can often improve the accuracy of your analysis. So it doesn't necessarily make life easier in terms of accuracy to take smaller data sets. There's also issues involved with just understanding the evolutionary processes that are going on. Adaptive evolution you can't really understand if you don't do a sufficiently dense taxon sampling. Now, over the last 10 years, we and others have developed many very good software products to do large-scale analysis. So the question could be perhaps moot at this point. Perhaps large data sets can be analyzed. Perhaps it's already done. Perhaps the big are easy. Some of you may remember there was a debate years ago about whether or not big trees were easy. I won't get into that debate just now, but I would say that it could be suggested that perhaps large data sets can be analyzed now and the problem is over. But I don't think so. In fact, what I would like to show you today is the progress we've made, but also how much is still to be done, and how, in fact, the more we know, the more we realize we've just started. And as you look at evolution in its complexity, you just realize that whatever you thought you knew, you have to revisit. And the computational problems, the statistical problems, everything gets harder and more interesting. So anyway, this talk is going to focus in 2 parts that will basically just feed right into, the first part feeds into the second, which is, the first part is progress so far on just a few topics, not on all of the topics we've been thinking about, but just a few of the topics. And then the problems that still need to be addressed. In fact, only some of the problems that still need to be addressed, because there are many. I just put down some of the ones that I think I could probably squeeze into an hour-long talk. So here goes. First, I'm going to talk about what we call absolute fast converging methods, which actually are a class of methods where you can actually prove something about the amount of data they need to be accurate with high probability. So that has to do with reconstructing an accurate phylogeny from sequences that are not too long. And what's the progress in terms of that kind of question? The second topic is improving heuristics for the hard optimization problems like parsimony and maximum likelihood. And the third topic is simultaneous estimation of trees and alignments. So the first 2 we know a lot more about. The third topic we know relatively little about, but we're starting. And then I'll just start talking about other things. Okay. So the steps in a phylogenetic analysis. Go out and you gather your data, and that could take a long time. I mean, especially if you're going out into the field instead of just downloading stuff from GenBank. Gathering your data can take a long time. Then you have to do a multiple sequence alignment. That step itself can take a long time and can require a lot of iteration between the biologist and software and data. Trimming the data, eliminating certain sites, eliminating certain markers, eliminating certain taxa, realigning, going back and forth like this. Okay, after the alignment is done, then you can feed it to a multiple sequence alignment method. That method may return many different trees. It could return all of the maximum parsimony trees. It could return all of the trees that are close to optimal for maximum likelihood. It could be a Bayesian analysis that's returning to you a sample of the trees. Whatever it is, it could return many different trees. From that collection of trees, you would like to establish what's reliable and what's not so reliable. At that point, you're applying probably some kind of statistical technique to estimate what you have confidence in. That's the next step. And then finally, you have an estimate of the tree with some confidence about certain aspects of it, and you try to say something about evolution. So that's the post-tree analysis step. So there are all these steps to this, and normally phylogeny research focuses just on the third step, which is given the alignment, find the tree. And I'm going to mostly focus about that during this talk. So this is a cartoon of a sequence evolving from the root of an evolutionary tree down the tree. And so when it's evolving with point mutations. So different nucleotides are being substituted by other nucleotides. And there's also this branching process. So that branching process just has to do with, speciation or duplication. So what you have is an evolutionary history relating the sequences at the leaves to the sequence at the root. And it's this branching process. But this is a very simple story. This is just saying everything is happening via point mutations. And then you can describe that process in terms of a statistical model and try to study estimation under the assumption that the data are generated by some kind of random process operating on that tree. So phylogeny reconstruction methods take the sequences that are at the leaves and estimate the tree. Now, the tree that generates the data is rooted, but the tree that you produce is unrooted. And so that's one of the limitations, is that tree estimation procedures don't actually produce roots. If you want to get a rooted tree, you include generally an outgroup, and then you can root the tree on the branch leading to the outgroup. But that's a way of handling the fact that the estimation procedures don't actually produce roots. Okay, so you get an unrooted tree back. And that's correct if it's the unrooted tree version of the true tree that generates the data. Okay, that's what we mean by a correct reconstruction. Now, of the various different reconstruction methods, there's basically three classes. The first class are heuristics that are trying to solve hard optimization problems. So that's heuristics for things like parsimony or maximum likelihood. And these combine generally hill climbing with some kind of randomness to get out of local optima. So these are NP-hard optimization problems, so that means that you can't actually guarantee that you're solving your problem on all inputs. And what you're hoping is that your heuristic is efficient enough that it won't get stuck in a local optima, and it will somehow manage to find good solutions, perhaps optimal solutions. And by optimal, I really mean global optima instead of local optima. Okay, so local optima are things like this. That's a local optimum. That means that if you want to find a tree of minimum cost, every tree that's nearby that point has a higher cost, so it's worse. But it's not a global optimum because this has a lower cost than this. Okay? So local optima and global optima are different concepts. The other class are the polynomial time methods, and almost all of these are distance-based methods. And what that means is that the first thing they do is estimate a distance matrix. And that distance matrix is supposed to be the evolutionary distance between every pair of organisms in your data set. These are fast. These can be highly accurate under some conditions, but they're not generally as favored as the heuristics for the hard optimization problems. And then the third class are the Bayesian methods that are generally using a random walk through model tree space to try to find a distribution that looks more or less like some distribution that relates to the data. So these are phylogeny reconstruction methods. And I'm mostly going to focus on the first two and say relatively little about the third. Okay, so the performance criteria. So as an algorithms person, if you want to evaluate the performance of a method, you can look at it with respect to various criteria. You can look at it with respect to how much time does it take, how much space does it use. You can look at it with respect to statistical issues. So if the data are generated by a random process and that random process has certain properties, you can talk about whether the method will be accurate given long enough sequences. You can talk about whether the method is going to be able to estimate the parameters of the evolutionary process, not just the tree. These are statistical performance issues. You can look at topological accuracy in simulation. You can, to some extent, look at that on real data, but then you actually have to know the correct tree. And that's not as likely on real data as it is in simulation. And finally, you can look at methods with respect to how well they solve optimization problems. And optimization problems could be things like maximum likelihood or maximum parsimony. And there you can talk about it on real data as well. Okay, this is the simplest of the Markov models that people have proposed for DNA sequences. And to make it simple, you're going to do everything that's obvious. You're going to say, well, first of all, the nucleotide at the root, you look at a single site, a single position evolving down the tree. And you just say, the nucleotide at the root is random. It's A, C, T, or G with equal probability. And then you just say, well, if it changes, if the nucleotide changes on an edge, it'll change with equal probability to the other nucleotides. That's a very simple assumption. And finally, you can just say that you just define the whole evolutionary process then as a rooted tree, and that on every edge you have a probability of change. And if you change on one edge, it doesn't influence what's happening elsewhere. So that's the Markovian assumption. That's the simplest model you could make up. And you could make this more complicated in all the obvious ways. You could have a distribution of nucleotides at the root. You could say that if you change, the probability of changing depends upon what nucleotide you're changing from and then to. So you can have a substitution matrix instead of just having it be the probabilities are all equal. So you can make it more complicated. But in general, all of these complications don't change the theory, as long as you keep them relatively nice. Okay. Now, this is just a single site evolving down the tree. How do the different sites relate to each other? Well, first of all, you assume that they're evolving independently of each other, because that makes it possible to do estimations. And secondly, you tend to assume that the sites are just scaled up or down versions of each other. So a site could be twice as fast as another, or half as fast as another, 3 times as fast as another. But basically, if it's twice as fast on one edge, it's twice as fast everywhere. So scaled up or down versions of each other. That's the rates across sites assumption. And then those rates you assume are drawn from some nice distribution. So that those nice distributions allow you to actually do the estimations. So that's a lot of what goes into these models, is keeping the models sufficiently simple and tractable that you can get provable guarantees about reconstructability. Okay? So here are 2 statistical concepts that some of you will know and some of you will not. And basically what we're saying, identifiability is a statement about a model. A model is identifiable if it's characterized by the probability distribution that it defines. Another way of putting this is that you can just say, if you look at one site and you look at how that site looks at the leaves of the tree, you know, is it all A's? Is it 2 A's and 2 C's? I mean, what does it look like? That probability distribution really tells you everything you need to know about the tree. Now, you might say it's enough if you can get the tree, but you don't get the substitution probabilities. But whatever what the aspects of the model are identifiable are the ones that you can get from those probability distributions. Now, that doesn't say anything specifically about a method. So a method is statistically consistent under the model if given essentially infinite data. In other words, given those, you know, the probability of the method reconstructing the correct tree goes to 1 as the sequence length increases. So that's a desirable property. It says you take more and more and more data, the method gets more and more accurate. It doesn't have to be monotonically getting more and more accurate, but it will get more and more accurate as the sequence length increases. So that's a desirable property. And if a method is not identifiable, if a model is not identifiable, then you can't have statistically consistent methods. So statistical consistency requires that the model be identifiable, okay? But having a model be identifiable doesn't mean that all the methods will be statistically consistent. Some may be, some won't be. Okay, identifiability results. There are increasingly more identifiability results. But the first ones are the easy ones, that the Jukes-Kanner model, the general Markov model, these are identifiable. So that means it's possible to get the tree back given infinite data. If the rates are drawn from a gamma distribution, and if you know the distribution, then you can actually get the tree as well. But check with Elizabeth Allman and John Rhodes for results that go beyond these things. They can tell you what's going on outside of those areas. Now, mixture models are an interesting thing. Mixture models are saying, suppose the tree's fixed, for example, but that the substitution probabilities depend upon the site. And so you have sites that are drawing with some probability from one model and some probability from another model. Those are mixture models. Now, what happens under mixture models is really complicated. And this is something where you can actually lose identifiability. In fact, you can get some pretty interesting problematic situations. And Mike Steele, who's here, can tell you about that. Okay, so identifiability is actually a very interesting research area, and a lot of what people would like to be able to do is unfortunately not going to work because the models that they'd like to estimate under are not identifiable. Okay? But check with these other people to get the latest updates on those. Now, some theoretical results about methods. Neighbor joining, which is one of the polynomial time distance-based methods, is polynomial time and statistically consistent under the nice models. Nice models like the general Markov model. Maximum parsimony, which is the problem of minimizing the total amount of change on the tree, is NP-hard. And even exact solutions to this NP-hard problem are not statistically consistent for some model trees, even under very simple models. Maximum likelihood is also NP-hard. But if you solve it exactly and you have a nice model, it will be statistically consistent. So these are some basic results. Not all of them known for a long time, but they are actually now established. And looking at this, you would say, well, obviously neighbor joining is the best. It's polynomial time and it's statistically consistent, right? Right? Good. But what about performance on finite data? I mean, after all, statistical consistency says what happens in the limit. As data get longer and longer and longer, how does your method work? What happens on finite data? What happens on sequences of length 500? What happens on sequences of length 50,000? You know, everything here is finite. What happens? How does performance work? So first of all, how do we quantify accuracy or error? So this is a cartoon of a simulation where you have a model tree. It generates DNA sequences, and then you take the DNA sequences, you feed it to your reconstruction method, and you get a tree. Again, this tree and this tree, this one's rooted, this one's not, but you can see that they're actually not the same tree, right? Modulo the root, they're still not the same tree, because, for example, this edge splits S4 and S5 from S1, 2, 3, right? There's no edge here which has that bipartition. So there is no edge corresponding to that edge in the true tree. There's no such edge in the estimated tree. Okay, so that's a missing edge or a false negative. Similarly, this edge, which splits S1, 2, and 4 from S3 and 5, doesn't correspond to anything in here, so it's called a false positive. So these are not the same tree. And the error rate when the trees are binary, the false positive error rate, the false negative error rate, they're the same. And so we're just going to talk about, in fact, either one of these error rates. In this case, there's two internal edges. One is right and one is wrong, so it's a 50% error rate. Okay? So the error rate we're going to be reporting is the percentage of the edges in the estimated tree that are wrong, and that's the same thing as the percentage of the edges in the true tree that are missing. Okay. So here's a simulation study that we did that shows as you take bigger and bigger and bigger and bigger and bigger model trees, and you simulate evolution on these model trees, but you fix the sequence length. So the sequence length here is fixed to 1,000 nucleotides. But we're taking trees from branch lengths from the same distribution, and we're just taking bigger and bigger trees. We start with 50 leaves, and we go up to 1,600, and the error rate's increasing. And then this is the proportion. So the largest it can be is 1. Okay, so when it gets to 1, you've missed every edge. So you're starting off mixing about 6% of the edges, but by the time you get to 400 sequences, you're missing 20% of the edges. When you get to 800, you're missing like 36%, and so forth. I mean, this is an error rate that is increasing. Now, for those of you who have done simulations, you can say the way that you're generating your model trees, as you're increasing the number of leaves, you're getting more and more homoplasy, because the tree is growing in its diameter, the evolutionary diameter. So more and more homoplasy means more and more back mutation and parallel evolution. And you're just going to lose the signal. I mean, you're not going to be able to reconstruct the tree. So what you're going to think is that this is a problem of the data, and that every method should have a problem with this data, and errors should just increase no matter what method you apply. But as you'll see, that's not the case. Okay? This is a condition that it's not unique to neighbor joining, but it's not a universal condition that applies to every possible method. So theoretical results about the sequence length requirement, again, how do methods perform on finite data? Neighbor joining and various other distance-based methods are guaranteed to return the true tree with high probability, provided that the sequence lengths are exponential in the maximum evolutionary distance in the data set. So that is a upper bound on the sequence length requirement, but in fact there is also a lower bound for a subclass of those model trees that will be shown later today. So there's matching upper and lower bounds for the sequence length requirements for neighbor joining, showing that an exponential sequence length requirement seems to be needed. And that isn't really what this is showing, but it's consistent with this. This is saying that as the evolutionary diameter grows, the error rate is growing. What the theorem says is that as the diameter grows, the sequence length you need for accuracy with high probability is increasing exponentially. So these are consistent trends, but they're not exactly saying the same thing. Okay, now what about maximum likelihood? Everyone thinks maximum likelihood is the best method on Earth, right? Not necessarily. Some people like parsimony, some people like neighbor joining, some people like Bayesian methods, but there's a big community that would say, sort of by definition, maximum likelihood should do the best. But the current best theoretical bound on the sequence length requirement for maximum likelihood is actually worse. So it's a real, we don't know really what's going on with maximum likelihood and its sequence length requirement. Ask Mike Steele about that. So this is a cartoon about what we mean by the sequence length requirement. So this is, you take one fixed model tree and you simulate evolution on this model tree. You take longer and longer sequences, and from each data set that you get, you feed it to a method, and you compute the tree, and you see how much error is in the tree, okay? And if the method is statistically consistent, then as the sequence length increases, eventually the error will go to 0, okay? That's with probability going to 1. Now, it doesn't have to go monotonically. It could be bumpy, but eventually it'll get down there. Now, if it goes down to 0 from a polynomial number of sites, then we will call that absolute fast converging. If it goes down to 0 from an exponential number of sites, we'll say it's exponentially converging. So that's how we're talking about methods. Methods that can go to 0 with high probability from a polynomial number of sites or from an exponential number of sites. Now, what I'm hiding here is polynomial in the number of taxa after we fix the other parameters of the model. So we fix the largest edge length, the shortest edge length, and what's left is just, we can talk about just the dependency on the number of leaves. So what we've said is that neighbor joining and various other methods, they're statistically consistent, but they seem to need an exponential number of sites. So does there exist methods that have this performance guarantee from a polynomial number of sites? And yes, the first ones were the short quartet methods of Peter Erdős et al., approximately 1995 and continuing. After that, we developed what we call DCM boosting for distance-based methods, and we got another collection of these absolute fast converging methods. Later, Elchanan Mosel, who's in the back, you can ask him about his work. Satish Rao and others have developed other AFC methods, which were based upon estimating ancestral sequences with high probability, and more recently, other methods as well by people in the audience. So there have been new AFC methods developed, and here's just an example of how one of them works. The same simulation I showed you earlier today, with neighbor joining's error rate increasing, this is what you get when you apply this technology DCM to neighbor joining, and its error rate is quite flat. So this is a technique where you're actually applying neighbor joining to subproblems and then putting trees together from subtrees. So it's a way of boosting the performance of the basic phylogeny reconstruction method. And we can prove theoretically that this method will converge the true tree from polynomial length sequences. So that's the power of one of these AFC methods. Nice theoretical performance showing that the problem is not the data, the problem is the method. So it's worth reconsidering what you think the limitations are of your data. Okay, but large data sets are probably better analyzed using other techniques, not neighbor joining, not DCM neighbor joining, but using techniques for either maximum likelihood or Bayesian techniques or even maximum parsimony. And we have been developing boosters like the one we did for neighbor joining, similar kinds of techniques, divide and conquer techniques, to speed up these heuristics for hard optimization problems. And here's an example of what we've gotten. So this is performance as a function of time, when you let time continue and you have a heuristic search for the best scoring tree. And this is an example of what you get when you're trying to solve parsimony. So over time, your parsimony scores are going to drop on your data set, right? Until eventually they converge, hopefully, to the optimal solution, the minimum cost solution. And this was a large data set with almost 14,000 sequences, and this was the TNT software improving over time. And this is the discovering boosted version of that same software and how it was dropping over time. So you can see a really big improvement in terms of the speed at which you got to a good solution. In fact, this would take, we believe, a few more months to get down there because we let it run for about 2 months and it hadn't gotten down there. Now that improvement in performance depends upon the data set. So when the data set is small, you don't get any improvement. When the data set is very hard and very big, then you get improvement. And we've also done this kind of DCM boosting for maximum likelihood as well. So we have these kinds of techniques for speeding up hard optimization problems. All well and good. And if that's all we needed to do, then we'd be done, right? We have fast ways of reconstructing large data sets, and we have nice theoretical guarantees about how much data we need. But evolution is more complicated than what we have described, okay? To start with, remember that second step in estimating a phylogeny? The second step is to align the sequences. All of what I've shown you has been assuming the data are already aligned. You already have a multiple sequence alignment. So what about that alignment, What happens there? Well, alignments are necessary because you have events like insertions and deletions. Not just substitutions or mutations, insertions and deletions. So for example, if you start with this sequence and then you delete this substring and change that T, you get this sequence. Now, insertions and deletions together are called indels, for insertions, deletions, indels. This is one event of length 4. It is not 4 separate insertions and deletions, indels. It's one indel event that at one point gets rid of 4 nucleotides. Okay? So it's one event. Now, what's the correct pairwise alignment on those two sequences? Well, you're going to want this A is the same as this A, right? This C is the same as this C, but this G has disappeared. So in fact, when you align it, you're going to put it over a dash. And these 4 things are going to all be over a dash, and then this C is going to be over that C, right? That's the sort of story you're going to get. So this is going to be the correct pairwise alignment right here. And if you have the evolutionary history on your tree and you trace it, on every edge you're going to have the sequences at the endpoints of the edge are going to be what you actually observed in evolution. And then the true alignment will be what you get by having this kind of a story going on. Then the true multiple alignment is the transitive closure of those pairwise alignments. So that's the definition of the true alignment, true multiple alignment, based upon pairwise alignments on the edges of a tree. So we wanted to understand how well the different alignment methods worked, and we did a simulation study. So remember, with doing a phylogeny estimation, the first thing you do is you take the unaligned data and you align it, and then you apply a phylogeny estimation to that aligned data. So what's the best way of doing that alignment, and what's the best way of estimating a tree given the aligned data? Well, we looked at various different alignment methods, including the ones that the protein alignment community had come up with that were performing very well on their data sets. And then we looked at maximum likelihood, we looked at maximum parsimony and various things. And we had, on this simulation study I'm going to show you, random model trees that all had 100 taxa. We had 2 different rates of evolution in terms of the substitution rate. We had 2 different gap length distributions. We had 2 different probabilities of gaps. So we have long gaps and short gaps, but everything's 100 taxa. And this is what we get. These are all maximum likelihood analyses of different alignments for the 8 different model conditions. And what you want to see as you go across is that within each model condition, in general, the relative order of the tree accuracy stays the same. So maximum likelihood on the true alignment, now we know the true alignment because we're doing a simulation, right? Maximum likelihood of the true alignment comes first, has the lowest error. Maximum likelihood of the prob-cons alignment and the mapped alignment are basically the next 2. And then maximum likelihood on clustol tends to be the 4th. Okay, except here. These 2 model conditions, somehow clustol is better than mapped and prob-cons. But in all of these cases, maximum likelihood on the true alignment is actually the best, which is not surprising. What's more interesting, however, is in some cases how little improvement you get over maximum likelihood on the true alignment. I'm not showing you here, but there's actually a lot of difference in alignment accuracy. But they're pretty close to maximum likelihood on the true alignment in a lot of these cases. Some of these cases you get big gaps in terms of gaps in performance. Okay, so what we're seeing here is that there are model conditions where you get a huge amount of error in your phylogeny. And if you could get a better alignment, you would actually do better, right? That's clear. Even with the true alignment, you're still missing 5% of the edges. So there's 2 problems with the 2-phase methods, and they each have to do with one of the phases. So there's the problem with the alignments, because the alignments can actually have a lot of error when you have a lot of gaps, okay? And that's one issue. The second issue is that even if you had the correct alignment, the reconstruction of the tree ignores the gaps. These gaps are treated either as missing data, so they're not taken into account, or they are treated as though each gap of length 10 is really 10 individual gaps of length 1. So that's a problem. You're either ignoring the gaps or you're overweighting them. So there's this problem with both steps, the alignment step and the phylogeny estimation step. What about simultaneous estimation? So there are Bayesian techniques and statistical techniques for estimating alignments in trees simultaneously, but they take too long. They don't actually scale. You can't run them on data sets of more than 20 sequences, and even 20 sequences could take you like a few weeks. So they're limited computationally. The extension of parsimony to this situation is computationally more tractable. There's software called POI. It's still an NP-hard problem, but it's more efficient in terms of its ability to analyze data sets. But it actually doesn't perform as well as the best 2-phase methods in most cases. So that's the current situation with simultaneous estimation. So we developed a new technique. And the basic idea of our technique is that we didn't want to have the sort of rigorous theoretical guarantees of doing right, because we wanted to scale, right? So we had to be able to define a heuristic approach to handling this problem that would give us better performance. So what we have is just a rather heuristic way of proposing multiple alignments. And then on each of these proposed multiple alignments, we will then compute maximum likelihood. But it's maximum likelihood under a model that doesn't include gaps. So it's really, again, a heuristic. Okay? We propose alignments. Each of these alignments, we then estimate maximum likelihood using RAXML for the software. Not published, but the same simulation, but our method now is the second method in this bunch of each of these model conditions. And in each of these cases, we're in between maximum likelihood on the true alignment and then the next two best. So you can see we're here, 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 and here. So remember, these were the two model conditions where maximum likelihood on the true alignment and the estimated alignments had a big jump in error, okay? And we are quite close to maximum likelihood on the true alignment in those cases. So we've actually really improved things. Alignment accuracy can be measured in a number of ways. This is one way of measuring it. It's the total number of columns. So it's basically saying, how long is your alignment? If the correct alignment had 1,000 sites and you estimated 1,100 sites, then you're 10% off. But if you estimated 2,000 sites, then you're 100% off. So this is normalized by the correct length, and what you can see is that in each of these conditions, we're quite close to the 100% mark. Slightly over here, slightly under here, but we're pretty close. Here we're actually more like 89%. But the other methods are really compressing the data, so they are over-aligning. Now this is something that has been observed by others, that most of the other techniques that I'm showing, in fact all of them, use progressive alignment. We don't use progressive alignment. Progressive alignment has a tendency to over-align. So that's an issue to be concerned with, is that when you're mistaking substitutions, or what is really a gap, you don't think is a gap, you think is a substitution, so you push things together. And that's a problem that the other methods are having, which might be one of the reasons that we're getting better topological accuracy, is we're not over-aligning. So for multiple alignment, Sate is giving us an advantage, but it's not sufficient. It's not doing that well yet. And so we still need to do better. But I would say that what our work shows is that trying to solve maximum likelihood with gaps as part of the process would actually probably give the best performance. So that's an open problem that I would like to pose, is how do we solve maximum likelihood under models that include gaps? So that's the first question that I'm throwing out there for people to solve. Okay, but that's still not enough, because evolution is still more complicated. There's genome scale evolution. There's a lot of interest in doing phylogenies on the whole genome scale now. And that means you have to be able to look at things like taking a genome where this represents one segment of the genome, this is another segment, and this segment is inverted. It's turned around, and so now it reads the other direction. Okay? Translocations move pieces from one place to another. So this is a translocation. Duplications take segments and invert, put them again next to each other. So that's, this piece gets duplicated and put right here. Okay? Now, how do you deal with all of these events? The alignment routines don't even work here, right? Because alignments just stretch things out and put them on top of each other. That won't work for inversions, it won't work for translocations, and duplications, it'll work, but it will treat this as though it was inserted instead of duplicated. This is still homologous to its ancestor. It won't show up as homologous in the alignment. So how do you even represent, how do you even do this kind of detection of homology from data? You can't do it using the standard alignment routines. How do you visualize it, and then how do you use it in a phylogenetic context? So here's a problem with visualization. I mean, this is a mess. I mean, this is the easy stuff to visualize. What about the harder stuff to visualize? You know, this is not easy to visualize. It's not easy to reconstruct. It's not easy to work with. So second class of things to think about for whole genome phylogenetics: given a collection of whole genomes, find the best alignment and phylogeny. That's a tall order. Suppose we make it a little bit simpler. We say we give you the alignment, so you know what's homologous to what. Now can you do it? Well, there's been limited progress there. If you ignore the substitution process and the insertion deletion process, and you're only looking at rearrangements, you're only looking at inversions and duplications and stuff like that, it's all NP hard, but there are to some extent some effective heuristics. But look how much we've had to ignore to be able to get to effective heuristics for small data sets. Okay? That's a big problem. So what happens if you allow yourself to work with all of those things at once? Big open problem. There's really just not enough done, even if the alignment is given. So if you try to do the alignment and do this, especially open. This is a big area with a lot of work that needs to be done. But evolution is still more complicated. There's the well-known distinction between gene trees and species trees. So this is just an example of how you can get it. It's not the only way you can get it. So you have this species phylogeny. This is where B and C are sister taxa, right? And A branches off first. But what we're tracing here is gene evolution. So you have a gene that's duplicated. So there's two copies, right? And now one copy is in red and one's in blue, and the blue copy disappears in the A lineage, but it continues down to C, and then it's on its way to B, but then it disappears also. Whereas the red copy doesn't even make it, it makes it partway into the C lineage and then disappears, makes it all the way into the B lineage and all the way into the A lineage. And so when you get around to doing the phylogeny, what you're looking at are the copies that you have accessible to you, okay? So you have the red copy from A, the red copy from B, and the blue copy from C. And when you do the phylogeny on those, you're going to actually think that A and C have a more recent common ancestor, because that's their ancestor, as opposed to the ancestor of all three, which is up here. So the gene tree is not going to be the species tree, okay? Now, you can try to get around this by saying, okay, we didn't pick the right markers, and you only look at other markers, but then you're throwing out data. So the question is, if you want to be able to use all of the data you have, you're going to end up with situations where your gene tree doesn't match the species tree. And you're going to have several different markers, and you're going to have different gene trees for each of those markers, and then you're going to try to come up with the species history. So people have looked at this problem, and in general, what they have tried to do is say, suppose you have the true gene tree for every single one of your markers. Then how do you come up with the species tree? And typically what they've also assumed is they have the species tree, but now they want to score it. They want to score like how much distance there is between each of the gene trees and that species tree. And that, of course, gives you a way of maybe selecting the best species tree, because if you can score each species tree, you could actually say which is the best. But the problem here is that we never get the true gene tree, right? Well, never is a strong statement, but rarely do we get a tree that we're absolutely 100% sure of, no lack of confidence. And so how do you incorporate uncertainty into this? Now, you could incorporate uncertainty into this by saying your tree is not fully resolved, or you could incorporate uncertainty by saying the edges have support estimates on them. But you have some kind of uncertainty. How do you incorporate that so that you actually are correctly evaluating the fit of a species hypothesis, a species tree hypothesis, to your gene tree hypotheses? And I don't think enough has been done in this area as well. So there's the gene tree, species tree reconciliation problem. But evolution is more complicated than this also. The tree of life is not a tree. Everyone knows that there's horizontal gene transfer. Not everyone knows about hybridization, where species get together when they're not supposed to, right? But that happens in fish, it happens in plants. It happens enough that you can get various kinds of non-tree-like models actually underlying your data. Horizontal gene transfer means you have extra edges on top of your tree. Hybrid speciation means that you actually have nodes with two parents, okay? So you really have to deal with the situation where your model of evolution is not a tree. And what do you do then? Some people would say don't even bother to find a tree. Some people would say try to find the underlying tree. But you know, whatever you do, you have to do something. There is a lot of previous work in this, NeighborNet, Splits Tree Network, and others. The main challenge here is that these methods produce graphs that represent the history, but what they're also representing somehow is the deviation from tree-likedness in the data. And many things can lead to a deviation from tree-like signal. You can have homoplasy, you can have finite data, you can have problems with your alignment. the model and the specification, distinctions between gene trees and species trees, all sorts of things can lead to this kind of non-tree-like signal in your data. So the big challenge here, again, is a statistical one, which is how do you look at these data sets and try to figure out how much of this is actually tree-like evolution, actually, with other things that are adding noise to the signal, as opposed to real reticulate evolution that's leading you to come up with some non-tree model. So it's not just a question of representing the noise in the data, but coming up with that scenario. So that's a really big area that needs a lot of good statistical work as well. Okay, but evolution is still more complicated. And it's just always more complicated than you think, and it actually makes a difference. So now we're going to go back to the simple things we talked about in the beginning, which is just modeling the variation between the characters or between the sites. There's all sorts of ways of modeling that variation, and heterotachy is one of them. Heterotachy just means variation in speed. But here I mean a very specific kind of variation where if this is one site evolving and this is the other site evolving, they have the same underlying tree, but the proportions of the branch lengths are not constant. So one is not just a sped up version of the other. So this happens. This really does happen in biological data. And when it happens, no matter how you define the rates across sites, you're not correctly modeling the evolutionary process because it's not a rates across sites hypothesis. So when you try to reconstruct trees from data which are evolving with heterotachy, your models generally won't fit. And what you really have is a kind of a mixture model going on. So mixture models, unfortunately, as I said, are not identifiable. So you have a situation where you're trying to reconstruct a tree from a situation which may not be identifiable, which means no matter what you do, you might have some error in your estimation, even if you do a very careful statistical analysis. So understanding the conditions under which the mixture models are identifiable or how much error will be introduced because it's a mixture model, this is something that we need to know more about. It's also computationally challenging to estimate under these models. So I think, John, you're going to talk about mixture models? Yes? And Simon Whelan is also going to talk about estimation under mixture models. So there are actually 2 more talks about mixture models coming up. So large phylogenies, they're important to estimate. They're important to estimate because with greater taxon sampling, you actually get insight into evolution in a way that you don't get without that level of taxon sampling. They are computationally very hard, however, and you have all sorts of issues that have to do with both statistics and data. In terms of statistics, with a very large tree, you're going to have more variation of models inside the tree. So, you know, as you move around the tree, there's going to be more models. You have to deal with mixture models there. You're going to have to deal, when you deal with a model-based approach, with lineage-specific and marker-specific variation. In terms of data, you're going to have missing data. You're going to have markers that only cover some set of taxa. Other markers cover other sets of taxa. You might need to think about what happens with supertree studies, but it's not clear that supertree studies are appropriate or that they'll work well. You have to deal with difficult alignment problems. You have to start thinking that things that you think are inaccessible to you that are fundamental aspects of the data, they may not be fundamental aspects of the data, they may be limitations of the current methods. So rather than saying we can't work with these data because we can't align them, we can't work with these data because they're too fast, we can't work with these data because they're too slow. I mean, whatever it is, maybe it's time to look at these data and say, rather than limiting our data that we're going to work with, let's characterize the data that's available and see if we can design methods that will work well on those data. So that's my preference, but other people will take a different approach. They'll say, no, those data are really a mess, let's work with a subset of the data. But then there are just these very specific issues that are like, you know, no matter what you do, no matter how good your gene tree estimation is, it may not match the species history, whether that species history is a tree or a reticulate phylogeny. And again, reticulate phylogenies are themselves huge problems. So problems we need to solve. This is sort of a summary of what I've had to say. Of some of the topics I think are really interesting, we also do need better supertree methods. Those are methods that take estimated subtrees and try to put them together into a tree on the full data set. It turns out that even the best of these supertree methods don't perform well enough. We need better visualization tools. We need visualization tools for large trees, and we need visualization tools for alignments, especially alignments where the process is not just insertions and deletions and substitutions, but where the process includes rearrangement events, duplication events, etc. We need visualization tools for reticulate phylogenies too. So those are things that we really need. But really, we need better models of evolution. And here I would say everything comes down to thinking about that, because we have learned so much as a community from doing both theoretical work in models and simulations in models. And yet, as we make the models more realistic, what we thought we knew changes. We thought parsimony was better than neighbor joining under these conditions, and now we see, oh my goodness, it's worse. Or we thought maximum likelihood was better than neighbor joining, or we thought neighbor joining was better than whatever. Even the relative performance can change as you make the models more realistic, as you make data sets bigger, as you make sequences longer or shorter. You make the models more realistic, your understanding changes. So we have got to have better models. And that's models for simulation purposes. Now, models for estimation may need to be simpler than the models we use for simulation. But we really do need to get as good an understanding as we can on what the molecular processes are. Not just so that we can understand phylogeny, but because that's really the driving interest in evolutionary reconstruction, is understanding evolution, right? It's not just what is the tree, but it's what is happening to life that's making it evolve. And then I have to thank my various collaborators, funding agencies, but especially the Newton Institute, which has put together an absolutely amazing program these last few months. And Mike Steele and Vince Moulton and Daniel Hewson, who have done an extraordinary job. Thank you.
Any questions?
Yes.
In your last slide before, you said we need better models of evolution. Now, I think we can't do that unless we try to include the changes of the environment that are either spontaneous, like some moon falling onto Earth, or even produced by life, like the first real serious pollution event that is much more serious than any global warming we see today, which was just the production of oxygen. It could have just finished life altogether, unless life would have been able to cope with the rising rates of oxygen. So if you really want to model evolution, just on sequence level or whatever, even less, or the distances of sequences even more abstract, you don't get to what's really happening.
That's a very good point. It's a very good point. And there are people, I don't actually know anyone who's doing that, but there are people who are using phylogenies to correlate events in evolutionary history with geological events.
They do that
but
in
what
New
you're
Zealand
talking
for
about is
sure.
even more interesting. Yeah.
Because they had this geological event of subduction. There was no New Zealand for many years.
And the rearrangement of the continents, et cetera. Yeah.
I didn't quite follow what you said about the SAT-E method, how it works. You choose the...
I didn't tell you how it works because I was speeding through this. SAT-E is actually sate, as in,
Right.
Thank you. So there should be an accent aigu over the E, but I don't know how to do PowerPoint with accents. So what we're actually doing is sort of doing a random walk through tree space, and every time we get to a new tree, we estimate an alignment on the tree by estimating ancestral sequences. That estimation of ancestral sequences produces a multiple alignment, and on that multiple alignment, we then estimate a maximum likelihood tree using RAXML, so GTR plus gamma plus I. So the real technique, the question about how this technique works has to do with how do we estimate alignments on trees, which is to say, how do we estimate ancestral sequences on trees, and then how do we move through tree space and alignment space, to do this? And that's a longer answer, which I can tell you during the coffee break. Yes?
Most of your analysis, or all of it, as far as I could tell, was based on the data just from the leaves.
Right.
Are there any, rapidly evolving species, microbes or whatever, whose genetic evolution can be studied to actually get a little bit of the history behind the leaves, or is that completely out of the question?
I don't think it's out of the question. There are these, people do grow things in their labs in order to do these kinds of studies. Yes?
I mean, all these viral sequences from
Excellent, yes.
From
Yeah.
grippe, what's that, in influenza, for instance, that Walter Pitch was analyzing are precisely data of that type. I mean, they change every year, influenza.
Right.
And HIV also. So you can do such studies, and they have been done.
And do they? I mean, so they could give you an idea of whether they are obeying a tree structure, or whether, because you've actually got some history for them. Do you know if that's the case?
In these studies, they just use maximum parsimony trees, and so
Right.
they
So they assume.
they construct a tree whether it is there or not.
And then there are other data sets where they actually do know the evolution, and they'd also know if it's reticulate because they can actually make it be reticulate. But those tend to be small data sets and not necessarily all that helpful in terms of evaluation of phylogenies. Yes, you had a question?
Yes, my question is about creating a new model to model evolution. Actually, what I think is, can we really think that it is really a challenge? I mean, with the data that we have, we can really think that we can build the best or more sophisticated model to model evolution. I mean, we have only a picture of the data that we have. We don't have a sampling of the evolution of the data. And without this, we can just make one-step model that typically are Markovian. So, how do you think which are for you the possible street that we could follow for?
So I think you're asking a pretty important question, which is to say, if you propose another model, how do you get a sense of whether this model is a better model? Not A, how do you come up with a new model, but B, if you propose a model, how do you get a sense about whether it's a better model? And there, it's a very interesting question, and Simon will show something about some models that he's developing and how they give better fits according to certain criteria. But these criteria don't necessarily give you the same scores, right? The Akaike information criterion and Bayesian, they don't necessarily give you the same measure of the improvement, right? But it's a very important question. I mean, one of the things that I think about in these areas is that we see certain kinds of properties when we analyze real data, right? And specifically when we're trying to solve optimization problems, there's a surface for the optimization problem that we can observe that doesn't fit when we get simulated data. Simulated data, you analyze very quickly generally. You get one optimal solution. When you take real data, you can find many optimal solutions. So, I mean, one thing is to sort of figure out the properties of the data you're trying to replicate and see if your simulations produce those properties. But I really don't have an answer to this. I think this is one of the important things that has to be figured out. How to come up with better models, how to test and see if those models actually are better, and to what extent do they actually make a difference in terms of your estimation. But there are people who are thinking about this, a lot, and that would be a good topic for one of the coffee breaks or one of the discussions.
Okay, thanks very much, Tandy. I think for the